plant telomeres in space and lunar regolith. Most of this work has been funded by NASA and by NIH, so thank you very much for everything. First of all, why do we need plants in space? Well, we're going to need them for food. Any space colonization event that is going to take longer than a couple of months in the International Space Station is going to require for us to bring the food with us. So we're going to need to bring plants that can be used as food, because we don't want to be eating ramen noodles for eternity if we colonize Mars or the moon. As well, we need them for oxygen generation, carbon dioxide removal, as well as mental health. Is one of the issues that nobody thinks about. When astronauts are in the International Space Station, one of their favorite experiments to do is plant-related experiments, and this is due to the fact that it reminds them of home. It reminds them of their backyard back home in Earth. So that is a very important issue, that plants do provide mental health support. And besides all of these very important reasons, in 5 billion years or in billions of years, the sun is going to explode. So if we want to survive as a species, we're going to need to make sure that plants can be used in interstellar travel and for colonization events. However, we have a huge problem. We don't really know if plants are long-lasting in space. So one solution that I'm going to be talking about today is to check their telomeres. So first of all, what are telomeres? So telomeres are DNA repeats, here in red, TTTA, GGG, which are at the end of the chromosome, and their function is the protection of the whole genetic material. Of special importance is an enzyme called telomerase, which is in charge of adding these telomere repeats at the end of the chromosomes. In addition to telomerase, there are a number of telomere-associated proteins, or TAPs, that are in charge of protecting the chromosome ends, the telomeres, from being recognized by nucleases that will lead to DNA damage response and eventually cell death. Every organism has a different telomere length set point, which maintains the rest of the genome protected. In Arabidopsis thaliana, the organism that I'm going to be talking about today, it is between 2 to 5 kb. Mouse, it is between 20 to 100 kb. And humans, we are between 5 and 15 kb. Mutations in either telomerase or telomere-associated proteins can lead to telomere shortening and telomere lengthening. So telomeres below the normal threshold of all of these organisms. Telomere shortening will lead to early senescence. And on the other side, telomere lengthening is one of the hallmarks of cancer. Organisms that have very short telomeres have lower chances of species prosperity. In addition to mutations in telomerase and telomere-associated proteins, there are a huge number of environmental and oxidative stress factors that can have an impact on telomere length and telomerase activity. Very interestingly, during my PhD work, I was able to test that plant telomeres are extremely resistant to environmental stresses, especially if we compare so with other organisms such as humans or C. elegans. So due to the nature of the repeats, TTTA, GGG, telomeres are hotspots of ROS, since these repeats are preferentially oxidized compared with the rest of the genome. Telomeres, as you can see, are composed of almost 50% of guanine bases that can be oxidized to 8-oxoguanine, which indicates genome oxidation and the presence of ROS at chromosome ends. As I just mentioned, environmental stress can lead to telomere length changes, and different levels of ROS will have a different impact on telomere length. So if we have a low level of ROS, we'll have normal telomere length within that range that is specific for every organism. However, as you increase the levels of ROS, you will have telomere elongation. And if you increase it almost to the maximum amount of ROS, this will lead to telomere shortening and eventually cell death. So due to the very important relationship between oxidative stress and telomeres, certain telomere-associated proteins and telomeres have been now found to have a role under oxidative stress. So telomerase has been found to move to mitochondria under oxidative stress to improve mitochondrial function. As well, its expression it is increased after stress. So I just want to point out that telomerase, besides having the critical role of maintaining chromosome ends, under oxidative stress has other functions away from telomeres and chromosome ends. So there's going to be two small parts to my talk. First is going to be the effect of low Earth orbit on plant telomeres, as well as beyond low Earth orbit studies that we perform. First of all, to study the effect of spaceflight on telomeres, NASA has performed two major experiments: the very famous twin study experiment and a 6-month astronaut mission. So experiments from the twin study, as well as the 6-month astronaut mission, revealed that astronauts, when they are in the International Space Station, they do suffer from telomere lengthening occurring the moment they arrive to the International Space Station. And this is independent of their duration. The moment the first studies were done, it revealed telomere lengthening, and this was as well further tested in the recent mission Inspiration 4, with which in hours there was telomere lengthening occurring on humans. One of the major hypotheses that there is for telomere lengthening, it is a potential telomerase activity. However, the experiment could not be due because the protein degraded upon return. As well, combined with all of this, astronauts experience an increased ROS, as well as genome and organellar instability. So the question that we had, it is, what is the effect of spaceflight on plant telomeres? Because remember, we need plants in interstellar travel for many different purposes. So the experimental setup, we prepared Arabidopsis thaliana seeds on MS Media at Kennedy Space Station. They launched on SpaceX resupply mission on June 3rd, 2021. And then seedlings grow on the veggie hardware that you can find right here with astronaut Thomas Pesquet inserting our experiment. And then Mark Van de Heide collected the seedlings, froze them, and they were returned here to Texas A&M, where we performed the experiments that we just recently, you will be able to read very recently in Nature Communications. So first of all, we wonder what is the effect, what is the status of plant telomere length aboard the International Space Station. For that, we measure telomere length on roots as well as shoots, because we know roots and shoots, they behave different in spaceflight conditions. We saw that compared with the control samples for both shoots and roots, you can see that the telomere lengthening with our TRF assay is the golden standard for telomere length assays in any organism. You can see that plant telomeres are between 2 and 5 kb, which is the normal range. Interestingly, we saw that flight samples, they were as well maintained within that 2 to 5 kb. We quantified this over here for easier interpretation. And we saw that there was no statistical changes in telomere length between flight samples for both shoots and roots compared with the control. So this is very remarkable, because this is the first organism that in a spaceflight does not have any changes in telomere length. These changes have been previously observed in humans and as well as C. elegans. So as I just mentioned to you in the introduction, telomerase, besides maintaining chromosome ends, it is very important as well for oxidative stress. This in plants remains still to be tested. However, we saw that if we look at telomerase activity with our TRAP and Q-TRAP assay, you see that there is a further increase in the activity of telomerase of space-flown samples compared with the ground control. This was quantified here for shoots and for roots. The change for shoots was around 4-fold change in relative telomerase activity of the space flown compared with the ground control, and a remarkable 150 average relative telomerase activity increase in space flown roots compared with the ground control. So we have a hypothesis that roots, unlike shoots, they're actually not adapted to deal with the damaging effects of radiation. And perhaps this is why we see such a remarkable change in telomerase activity in roots versus shoots. Very interestingly, as you just saw, telomere length did not change in space-flown material. However, the telomerase activity, it is tremendously increased. So why is this happening? Why is telomerase activity being increased? And I remind you that this is the first telomerase activity assays that have been performed in any space-flown organisms. We also have previously tested, I'm not going to show it over here, that other environmental stressors have been shown to lead to telomerase activity upregulation in plants without changes in telomere length. Different studies have tested the effect of spaceflight on different plants, and one of the most common responses across all experiments and across all organisms, it is the increase in oxidative stress. So compared with ground control, we observe an increase in atoxoguanine. Remember, atoxoguanine, it is genome oxidation. So there is an increase of oxidative stress that has a damaging effect on the genome, as is what tested for both shoots and roots of spaceflight versus the ground control. And in addition, and very similar to humans, there was changes to organellar morphology. We observed that the DNA content of spaceflights, the mitochondrial and chloroplast DNA content was increased, meaning that there's an increased amount of both mitochondria and chloroplast on a spaceflight tissue. So there are many different stressors that are happening on the International Space Station, but the most important ones probably are the effects of microgravity and the effects of radiation. So first, we had to understand whether these phenotypes that we observe are due to radiation or microgravity. So we went to Kennedy Space Center and we kept plants on an RPM machine to mimic the effects of microgravity. So plants were rotated in the RPM that you can see over here for 12 days, and we studied the effect that microgravity alone had on plant telomeres and telomeres. I like to say that the RPM keeps the plants very dizzy for 12 days. We observed, the analysis revealed that telomere length remain unchanged, similarly to spaceflight, so no surprise here. Telomeres remain stable with microgravity alone. However, and very importantly, we observed that the telomerase activity remained unchanged for our RPM samples compared with our ground control. There was a slight increase in telomerase activity in roots to 2.5-fold compared with the control. But remember, spaceflight roots had a regulation of 150-fold change in telomerase activity, meaning that probably telomerase activity that we have observed could be due to space radiation effects. So are plants better equipped for space travel than humans? Based on these studies, as I just showed you, astronauts experience an increase in telomere length independent of the duration. We do not know whether that's due to an increase in telomerase activity. However, there was an increase in oxidative stress as well as mitochondria. Plants' telomere length remained unchanged. However, there was a massive increase in telomerase activity that is potentially due to the increase in oxidative stress. By the end of the year, we're going to go back to the International Space Station, where we're going to study telomerase-associated mutants in the International Space Station, as well as perform further radiation experiments on Arabidopsis thaliana plants to mimic the amount of radiation they will experience on a long trip to Mars. So the first part, I showed you that plants on low Earth orbit, they suffer from increased genome oxidation. Telomere length remains unchanged. However, a massive increase in telomerase activity. So on part number 2, we're going to study what are the effects of taking the plants beyond low Earth orbit to their telomeres and telomerase. So we are all aware of the recent experiments undertaken by Annalisa Paul and Robert Fair of growing Arabidopsis in genuine lunar regolith. These plants displayed increased ROS, as it was evidenced in their transcriptome studies. The question that we have is, can plants live on the moon for many generations? And for that, we're going to be looking at different physiological and molecular markers of fitness, with obviously a special look at telomeres for different generations of Arabidopsis grown on the simulant regolith. So first of all, we grew Arabidopsis with the simulant Lunar Mar S1, which we obtained from Exolith Lab. And we just look at germination of Arabidopsis. As you can see, compared with the earth soil, plants that grow on their LMS1 untreated regolith, they were not very happy. They were pretty much died after a couple weeks and completely dead by 1 month. However, as I just mentioned to you, Annalisa Paul tested that there was an increase in oxidative stress in plants growth in the regolith. So what we did, it is treat the regolith itself with an antioxidant cocktail. And this led to a massive increase in germination potential. As well, the plants, they were able to fully develop and go through all their life cycles. We quantified the rosette area of the earth control antioxidant and untreated. And as you can see, the untreated, it was very bad, untreated regolith. However, with antioxidant, we're able to make the plant happier, so to speak. It will go through different generations, but there's still a difference in size, biomass, rosette area compared with the earth-grown plants. So we next proceed to check the generational growth of these plants. And compared to G1, you can see that there's a further decrease in the rosette area of G1 here in blue, on G2 here in blue compared with the G1 here in pink. And as well as the biomass was decreased. Here is the picture of those plants. So there is a generational stress that is being maintained with the subsequent growth of plants on the lunar regolith. So the next we tested to see how the genome is doing, because the genome will really tell us if there can be more generations after G2, or if we really have to worry about this plant's generation now. So we then look at telomere length changes, and we observe that G2 plants here with our TRF assay, they suffer from a further shortening of telomeres compared with the G1 and Earth. We quantify this over here, and there is a consistent loss of around 500 base pairs between G2, G1, and Earth control. So this is telling us already that these plants will probably not be viable for many more generations, and they're eventually going to die. We further tested with another telomere-related assay and saw the same effect, that G2 lunar plants have a further shortening compared with the Earth control plants. And this is bad news. This means that plants will not be able to grow during multiple generations using the lunar regolith. So what is the reasoning of these telomere length changes? Because if we know why these telomeres are getting shorter, perhaps we can apply that to the regolith or genetic engineer the plants ourselves to make the plants sustainable for different generations. And we observe that plants that grew in the regolith, they have an increase in atoxo-G. Remember, this is genome oxidation, a massive increase in genome oxidation. So these plants, their genome, it is getting completely oxidized. And this is probably why they will not be viable through different generations. We as well saw that there's a further loss of the genome integrity, which is consistent with genome oxidation. As you can see, we tested the amount of fluorescence that is coming from the chromatin, and we observed that the pixel intensity, meaning the chromatin compaction, it is further decreased in the lunar grown sample. So the genome, it is, as we are seeing over here, completely getting trashed. Very interestingly, why do we see that telomere shortening? Is it because one possible explanation is that there is oxidative stress that is damaging the telomeres themselves. The other reason that could be is a telomerase activity changes. And we saw that even G1 lunar plants displayed a decreased telomerase activity compared with Earth control. So what we have hypothesized over here, it is plants that are growing on the moon, they are suffering from oxidative stress that is leading to telomere length changes, and this is being combined with telomerase activity being downregulated. So you keep seeing that loss of telomere lengthening that eventually is going to lead to non-viable plants on the moon. So to put all of this into perspective, all of the research that I've talked with today, plants on the moon, they suffer from telomere length shortening, decrease in telomere activity, and a massive increase in oxidative stress. We obviously don't know what happens to humans on the moon, but this while plants are able to live successfully in low Earth orbit compared with humans, based on the data that I just showed you, plants grown on regolith, they're going to struggle. So we're going to need to figure out different genetic modifications for these plants to be able to be used for lunar and Martian colonization attempts. So putting all of this into perspective, I hope I was able to convince you today that telomeres are markers of cell and organism prosperity. Spaceflight did not lead to changes in telomere length, but telomeres activity was greatly increased, probably due to oxidative stress coming from radiation. Plants grown on lunar regolith display physiological and molecular stress with shortening of telomeres in second generation. Similar phenotypes were observed on Martian regolith, but more dramatic. I did not have a chance to speak about this today. And I just want to mention really quick, just for the sake of time, that antioxidant and telomeric genetic enhancement of plants shows promise on lunar growth. And this is new data that is coming from the CPEN lab, that we have some plants that they have been genetically engineered to be able to grow on the lunar regolith without having these phenotypes that I just showed you about. So thanks again for the CPEN lab collaborators, Dr. Wyatt, Emma Canady, Dr. Pereira, Eric Land, Dr. Roberto Arquilano, Dr. Susan Bailey, that she is the twin study main principal investigator. NASA Plant Analysis Working Group, as well as Dr. Meyer and Jeffrey Richards. And thanks again for funded by NASA and NIH. And thank you very much for everything, especially for Robin and Dan. You guys are amazing people. Thank you.